All right. Well, welcome to the webinar. And my name is Jing Li. I'm the Director of Life Sciences and Technology at GemPharmatech. I have my colleague Steve Smith on with me. Steve is the speaker for today's webinar. So without further ado, I'm going to give a short introduction to Steve, and then Steve will share the webinar with us right after. So Steve is a research scientist and a business development professional with over 2 decades of experience in biotech drug discovery and business development. He has designed and conducted research in therapeutic areas spanning oncology, metabolic diseases, neurodegenerative diseases, inflammation, and immunology. He has also built in vivo and in vitro systems for disease model research. Prior to GemPharmatech, Steve has led the business development as Gliologics, which was acquired by Zogenix. And then he returned to the bench to lead the development of models for exploring new therapeutics. Steve has a PhD and a Master's of Science in Microbiology from North Carolina State University. His bachelor's degree is in biology from Baylor College. Today, Steve is going to share with us the most recent data on one of the newest disease models we have developed. Is the FAD4T, is a mouse model for Alzheimer's and other neurodegenerative diseases. All right, Steve, the stage is yours.
Great. Well, thank you so much, Jing. I really appreciate it, and look forward to telling the group about our about GemPharmatech's development with Alzheimer's disease models. So let me go to the full screen here. Okay, great. Thank you. With GemPharmatech, we have a very large collection of genetically modified mouse models, and in particular, we're for this audience and in my scientific interest, we have quite a few models of neurodegenerative disease, including the newly developed FAD4T model that I'll be discussing today. And so the disclaimer that we always have to show, legal disclaimer. Alzheimer's disease, very important and crucial disease, primarily affecting older, middle-aged, late middle-aged elderly people. And Alzheimer's disease was identified in 1901 by Dr. Alzheimer, who examined a patient who had dementia. And the Dr. Alzheimer used cell staining techniques to identify the amyloid plaques and neurofibrillary tangles within the cortical gray matter of the brain, which is the hallmark, of the disease. In 1906, with his continued pathological research, he linked these pathologic findings with the clinical symptoms of presenile dementia. Alzheimer's disease is a central neurodegenerative disease with plaque accumulation, tangles, and a shrunken brain. In 2015, there were over 46 million people with Alzheimer's disease worldwide, more than the population of Spain, as an example. The number of human patients is estimated to increase by nearly threefold by the year 2050. The total estimated worldwide cost of dementia in 2015 was over $800 billion, becoming a $1 trillion disease by 2018 and anticipated to rise to over $2 trillion by the year 2030. If dementia care were a country, it would be the world's 18th largest economy, more than the market values of many very large successful companies such as Apple and Google and others. Alzheimer's disease is the most common form of dementia, and it does not have an effective treatment at this point. As symptoms worsen, individuals must rely on caregivers for their daily needs, resulting in serious social as well as financial burden. The graph on the left you see right now shows the increase in costs on patients and their families due to neurodegenerative diseases, nearly tripling from 2010 to 2030. On the right now, you see that you can see that the single largest cause of neurological disease is Alzheimer's, of all of the various neurodegenerative diseases, followed by traumatic brain injury, stroke, epilepsy, and chronic pain, and Parkinson's. Okay. There are many hypotheses regarding the real drivers of Alzheimer's disease, including abnormal deposits of amyloid beta protein in the extracellular spaces between neurons, formation of tangled tau protein fibers inside the neurons, cholinergic neuronal damage, inflammation, and oxidative stress. Many of the Alzheimer's disease models are based on the A beta hypothesis. GemPharmatech has also developed models to test this as well. There are primary drivers that are known drivers of Alzheimer's disease, including mutations in APP, PSEN1 and 2, ApoE4, and TREM2, but also many other mutations and mutant loci that are involved in disease. APP has amyloidogenic and non-amyloidogenic pathways that it's involved in the body. In non-amyloidogenic pathways, soluble APP alpha is generated from the cleavage of APP by alpha secretase. Here we go. And it shows biological functions in growth regulation and neuroprotection. In amyloidogenic pathways, APP is cleaved by beta secretase, base 1, and gamma secretase, forming A beta. In a healthy brain, the A beta 40 peptide is degraded, but in Alzheimer's patients, the A beta 42 peptide fragments accumulate, forming insoluble plaques. And these plaques, well, totally insoluble, and they catalyze formation of additional molecules coming together. In patients with many of these mutations, here on the right, you can see within the APP, there's mutations known as the Swedish mutation at 670, 671, then also Florida mutation, London, and Indiana mutations. And in humans, these are the most common mutations driving disease. More than 200 Alzheimer's disease therapeutics have been or are currently in the clinic. Shown here in this table, there's currently about, this is fairly current data. Within the last 5 years, there were over 100 drugs and drug targets. Primary targets are A beta, tau, base 1, and gamma secretase. However, few drugs make it to phase 3 or even the market, suggesting that there are more appropriate preclinical models that should be developed, need to be developed in order to study and treat this disease. Animal models of Alzheimer's disease are valuable tools to understand familial or early onset Alzheimer's disease. There are several classical Alzheimer's disease, such as several classical models, including the APP presenilin, the 3X transgenic, and the 5X FAD, or familial Alzheimer's disease. These various models have a range of and combination of human mutations resulting in disease in the mouse models. So GemPharmatech realized some of the pros and cons of these mouse models, and with that knowledge and with the ability to efficiently generate genetically modified mouse models with precise mutational strategy, they've generated the FAD4T mouse model, and to really explore Alzheimer's disease in new directions. With this model, So as you might guess, there are 4 mutations. 2 of these transgenic mutations, Knockian mutations, are in the APP, using the Swedish and Indiana mutations. And in PSEN1, there's also 2 mutations. This model has several advantages, including it develops A-beta deposits by 6 months of age, and A-beta deposits at 4 months of age are comparable to the 5X FAD model. It's also generated by GemPharmatech and has independent IP. The sales are not limited. If a researcher is interested in the model, they can purchase the model from us. We can breed up the animal model and send animals to them. We can also internally conduct studies at our facilities in China, as well as partners here in North America. And there's a favorable price to this model too, making it easier for not only academics, but industry groups as well to use and explore this model in their research. Using an antibody that specifically reacts with A-beta plaques, we detected traces of A-beta deposition as early as 1.5 months in the FAD4T mice, with A-beta deposition increasing gradually with age, as you can see here in these images. The A-beta plaques are found to be more severe in female mice than in males, which is consistent with clinical pathology in humans. This figure shows data using female mice and their A-beta plaques. Whoops. The data from collaborators of ours with this mouse model shows that when compared to the 5X FAD on the bottom, that A-beta plaques in the cortex and hippocampus of 4-month-old FAD4T mice is comparable to that of similar age 5X FAD mice. Astrocyte activation was detected in FAD4T mice at 2.5 months of age, and significant glial hypoplasia was observed at 7 months of age. The novel object recognition task is based on the natural tendency of mice to investigate novel objects instead of a familiar one. Mice were first given 20-minute habituation with no objects in the open field, and then a test phase consisting of 2 trials beginning 24 and 48 hours later. In the first trial, 2 different objects were placed in opposite corners of the open field. And then the mice were allowed to explore them for 10 minutes. And then 24 hours later, one of the objects was replaced with a novel object, and a mouse was again placed in the maze for 10 minutes. The mice either explored the novel objects for a longer time, indicating memory of the familiar object, or exploration of the novel and familiar objects for the same length of time, indicating a lack of recall, lack of curiosity, or lack of memory for the familiar object present during the initial phase. So you can see that there were slight changes in the animals, with a bit more of a disagreement between wild type and the FAD4T animals in the female mice. The Morris-Water maze test, it was used to measure spatial learning and memory. The maze is equally divided into 4 quadrants with 1 hidden platform. The training sessions were conducted in 4 consecutive days with 4 trials per day, followed by a probe test on the 5th day. In each training session, mice were randomly released into the 4 quadrants and allowed to have 90 seconds for finding the hidden platform and 30 seconds to rest on the platform. And then if mice didn't find the platform within 90 seconds, we guided to the platform where it stayed for 30 seconds. For 3-month-old mice, the FAD4T mice showed natural learning and memory abilities. compared with wild type mice. And that's shown here in figures A and B. While in the spatial search experiment, there's a trend toward cognitive decline in FAD4T mice, female mice, compared with wild type mice. As you can see here, some disagreement, indicating by less transition numbers, less time spent in the target quadrant, and that is shown in figures C and D here. Again, another continued work with a collaborator. They found that mice that were subjected to a learning paradigm of the water maze, Morse water maze for 6 days, followed by a probe trial on day 7. In each training session trial, mice were randomly released into the water from 2 quadrants of the pool and allowed to have 60 seconds for finding the hidden platform, 15 seconds to rest. As shown in the learning sessions, the female and male mice displayed significantly longer latency, indicating the FAD4T mice were impaired in their spatial learning capacity. In the probe test, the number of crossing platforms was decreased significantly in the female FAD4T mice. These results indicate 5-month-old FAD4T mice have spatial learning and memory impairment. Cognitive deficits were observed in FAD4T mice in both males and females, and with the actual moving distances, and this is conducted using an open field test. In the moving distances, the wild type and the FAD males had similar moving distances as females, and the velocity, the speed at which they're moving when they're roaming around, was similar. However, the time spent in the center, which shows that they are they do not possibly have fear conditioning, is very different in the both males and females, and the time in the center area, again, so time in the center was similar, or was dissimilar in the FADs. In a test looking at therapeutic efficacy of a experimental drug that's in the animals, tests were done at a low, medium, and high dose. So there's, as you see on the x-axis, each examination looking at over time, over training time, that the animals were not, regardless of how much the treatment was, they were not reaching near what the control wild type animals were as far as escape in their movement in their cage. Interestingly, there appeared to be some slight improvements in the low dose treated animals compared to the other FAD4T animals. However, this was not observed in all parameters or in all animals. There is still a fair amount of variability seen within even the reasonably successful group. In this table compares the disease physiology of the FAD4T mice with the APP PS1 animals, the 3X transgenics, and the 5X FAD models. Just looking at the deposition of A beta plaque, the neurofibrillary tangles, behavior with the mutations that are present that drive disease in the models. Next, be discussing the Alzheimer's disease plus mouse model. Chromosome 1 consomic strain, and with and these are new models to explore Alzheimer's disease pathology and other neurodegenerative diseases, and explore therapeutic efficacy with these mouse models. The Alzheimer's disease, the 80+ mouse model, just looking at what is it in the human condition that makes Alzheimer's disease, despite having the same primary driver mutations, why is the disease so much different in one patient versus another? And so just looking around at known genome-wide variation, SNP analysis, and where those are, what genes they are, has had us just looking at, from a basic research standpoint, and considering what we could possibly do to create a new model, unique model that might be better representative of human disease. And so our goal, develop an improved mouse model for preclinical research, and we started constructing a consomic mouse strain of consomic chromosome 1, consomic 1, CSS. And with that, bred with a wild mouse strain, the donor strain, and we selected animals through many rounds of breeding, selected animals that had chromosome 1 from a wild strain and were otherwise inbred mouse strain. Upon initial examination of these animals, you can see that they have somewhat improved movement for both the males on the left, and females not so different on the right, with the consomic strain versus the wild strain. When looking in the Morse water maze, looking at platform location crosses, there was some difference in the males. In the time spent in quadrants, again, there was some difference in the males, and the actual speed was about the same for both the consomic strain males and females with their wild type counterparts. When this consomic strain was crossed with the 5X FAD mouse model, and tests were run, interestingly, we saw that the 5X FAD with the chromosome 1 consomic from wild mice actually performed a little bit worse than the wild type animals, and worse than the consomics on their own. So some combination of the 5X FAD and the wild chromosome 1 gene variants altered the behavior and activity in those animals. We also saw that with these mouse models that they have some spatial memory deficits that are greater than in the 5X FAD alone, and the chromosome 1 mice, the 765 animals, are actually, as on their own, very low absence in Alzheimer's disease plaques. And so for some reason, the combination of the 5X FAD along with the consomic chromosome 1 resulted in slightly worse disease, which became statistically significant upon histological as well as behavioral analyses. And this slide is showing that within GemPharmatech, we have a wide range of research models that are in development or have been developed for a wide range of neurodegenerative diseases, including prion diseases, Alzheimer's, Parkinson's, ALS, Huntington's disease, and Lewy body disorder, and FTD. And those with the stars, those have mouse models that are currently developed or are in development. GemPharmatech has also developed a wide range of humanized mouse models of CNS disease. And many of these are being released, or they will be released very soon. And with these, we hope to work with outside groups to help characterize the mouse models for their research, for their own internal research studies, but also if we could work with groups who could characterize the mouse models for our own, for better understanding the mouse models themselves, that would help us and our marketing team. And we would love to work with you, anyone who is interested in working on these or any of our other neurodegenerative disease mouse models, to bring them forward and bring them out into the research market. We also have other models that, as I mentioned, that have been developed or again are coming forward, and these being released or have been released and currently available, including the FAD4Ts. That concludes my discussion for today. And I'd be very happy to hear what you hear your thoughts, your interests, any questions that you may have. Thank you very much.
All right, thank you, Steve. And I'll answer the first question. So we got a couple questions in the chat box, a couple questions in the Q&A. I'll repeat the first question and I'll answer that so you can take a little break. So the first question is, will the slides presented at the webinar be available after the talk to the attendees? The answer is yes, but you are highly encouraged to email us so that we know who to send it to after the event. And so Steve, I'll read the second question to you. What is the donor strain of the origin of chromosome 1 in the consomic mice?
The donor, I am pretty certain that it is a wild mouse strain, and I'm not sure where that wild strain came from, but that was, I think that may have been on a slide that was deleted from today's presentation.
Yeah, I can chime in
Yeah.
To help Steve out. I did, we have another webinar before that we also mentioned the consomic strain as one of our methodology going forward to generate new models for disease exploration. So the consomic strain, these are really, as Steve said, wild animals we caught from the wild. This particular one is, I believe, in China, Yunnan province, if you're familiar with the geography in China. All right, next question. What are the differences between the FAD4T model versus the 5X FAD? I think you mentioned that during your webinar. If you don't mind, can you, just highlight a couple of points that you want to share with the audience again?
It's many of the same mutations in the 5X as well as the FAD4T models. And it's really that the FAD4T has two mutations in APP, the Swedish mutation and Indiana mutations, whereas, and then in the PSEN1, they've got two very specific mutations in there. And so it's similar to the 5X, but the 5X also has the Florida and the Swedish mutations, and one of the same PSEN mutations.
All right. Second one I saw from the Q&A for the Consomic 765 model. It is still unknown what caused the difference between the 765 versus the wild type mouse.
Yes, that's still unknown. We're doing some work internally. In China, we have collaborations with, I believe, 2 academic groups that are exploring those differences. And so we're very excited about working with any other groups to help explore this as well. We know the value of doing some genomic profiling, and we just don't have the bandwidth to approach that. But if some group would like to work with us on this, we would really enjoy working and learning about the model more. Thank you.
Yeah, fabulous. So I'm now coming back to the chat box. I got your email, Alexander. We'll definitely make sure to send the PowerPoint to you after the event. I also see another question. Do any of your mouse models exhibit increased oxidative stress?
We do have some, but within the KOAP library, there are certainly some models that are designed to explore, for instance, mitochondrial genes or proteins that are important in mitochondrial function. And those could be important for looking at oxidative stress. And then there's a whole host of other models within the KOAP library for looking at oxidative stress and its ties to inflammation. For these neurodegenerative models, we haven't generated any specifically for that purpose, but I'm certain that there are some that would have great application for that.
Mm-hmm. I think this question seems to, tag into the phenotyping of the models. So Steve, you want to share some, more information regarding model co-development that we do?
Well, yes, I mean, that's an excellent point that we have certain things that, we have a battery of tests that we're conducting on our mouse models, but we can't obviously come up with the plan for running every study that's out there. And so if you would like to work with us and conduct some specific studies, because you're probably more of an expert at these oxidative stress assays, that would be wonderful. And we would be very happy to work with you. And please email us, contact us, and we'll discuss supplying sufficient animals for you to get going with your research.
Great, thank you, Steve. I see a question. I'll read it out to you, and I think we can probably both share some perspective on that. So the question is, are there history slides that are available for immune studies?
History slides?
I'm guessing, so, Paul, you can text more in the chat box to let us know. I'm guessing your overview at the beginning or your review at the beginning for the neurodegenerative disease area, I think Paul is probably—
Oh, histology, okay. Yeah, I can send, with the slide deck, I can send histology, and we have extra slides that didn't make it to today's presentation for histology as well as behavioral assays that I would be happy to share with you and discuss further assays that we've conducted.
But still, do we have histology slides that are available for immune studies?
Oh, I don't believe we have histology slides that are currently available, but we could make some available for you since we have the colony. If that's what you need is just histology, conduct histology, we could send you frozen samples that you could, or preserved samples that you could then stain later, or we could work with you and have, with our mouse models, we could collect some tissue and stain for you.
Yeah, so Paul put in the chat box, yes, for target validation. So I think this is really pivoting into a model validation,
Yeah, I see
model
that.
establishment. So Paul, highly recommend that you let us know your email. You can, put in the chat box as Alexander did, or you can, offline, use our contact us on the website to let us know you talked with us and you want to have further communication regarding the histology analysis with us, and we'll respond to you accordingly. All right, I'll check one more time in the Q&A. Okay, there's one more saying, did you compare weights over time or record grooming behaviors or locomotion in the cage? Any sleep recording?
Yeah, there, in one of the slides or a couple of slides, there was locomotion, just looking at the speed that animals moved. I'm pretty sure that we have information on distance traveled as well. So time spent moving, distance traveled, and speed. And in slide I showed, not all of that information could be summarized. And I could send that information to you if you would like that. And I don't know if body weights were captured. In my experience, I certainly would have captured body weights, and I assume that our research team did as well, but I'm not sure of that off the top of my head. I can check on that, or other behaviors. I see grooming, that's definitely something that changes from model to model, and I don't know if that has been qualified or quantified in these animals. I can inquire about that internally, however.
Yeah, and I guess sleep recording probably we wouldn't have had, but
I doubt it.
again, yeah, but again, we can inquire with the R&D team and make sure we don't miss information. Yeah, so let us know or contact us after the event. Right, checking one more time. I don't see any more questions, so with that, I will say we'll conclude today's webinar, and thank you again for joining us, and I will see you next time.
Thank you very much for attending. I appreciate it. Thank you for your interest.
Bye-bye, everybody.
Bye-bye.